So today's seminar speaker is Dr. Jessica Ware. She's an alumnus from our department. In fact, she and Dr. Nielsen graduated at the same time. And when I started in 2007, definitely Dr. Nielsen and Dr. Ware were like trying to get them, come on, set up your lab so we can do some molecular work. This is interesting. I'm kind of being hounded by graduate students. She is now the curator of the American Museum of Natural History, where she serves as the chair of the Division of Invertebrate Zoology. She research focused on the evolution and behavioral and physiological adaptations in insects, with an emphasis on how these occur in Donata in particular, and also Dictyoptera, so the nice termites and cockroaches. She holds up, I did not know this, you're Canadian. Yes.
Yes. How do you not know that?
From Toronto.
It's like the first word I've ever heard.
She speaks English.
Mais je peux parler français aussi.
She got her bachelor's at the University of British Columbia and then her PhD, as I mentioned, here at Rutgers. She was awarded the Presidential Early Career Award for Scientists and Engineers from the US government for her work on insect evolution, which we need to find out how we do that. I mean, besides being spectacular, which is probably the best way to do this. So we got a Presidential Early Career Award on insect evolution. That's really cool. You served on the governing board of the Entomological Society of America for 3 years. She was the director of diversity, equity, and inclusion at the Society of Systematic Biologists for 3 years. She's past president of the Worldwide Dragonfly Association, great name. Let's go worldwide, and the past president of the Entomological Society of America. She's president-elect of the Society of Systematic Biologists and the co-founder of Entomologists of Color, and serves on the board of Black in Natural History Museums. It's my pleasure to introduce Dr. Jessica Ware.
Thank you so much, Dr. Fonseca. I'm really honored and humbled to be back here. When I did my PhD, we were in Blake Hall, but it's nice to see so many familiar faces. And I'm honored to be here to be speaking in the Marmarash seminar. When I first started, I came from Canada. See you later, Canada, I said. I'm moving to the States. And a lot of my Canadian family thought that was like not a great idea. And Dr. Marmarash, his office was kind of down the hall from where my office was initially. And we talked a lot about coming to this country in pursuit of science. And he was always just a source of optimism. And he had really great pride in insect physiology work. And he was a real inspiration. So I'm honored to be able to be speaking in a seminar that has his name. And so I was here with Dr. Nielsen, as was mentioned, over in Blake Hall. And I also had Dr. Hamilton serve on my thesis committee. And it was really a great opportunity to be here in the Department of Entomology, a really old entomology department in the history of this country. We felt proud to be here. But importantly, it was a great chance to form lifelong collaborations and friendships. And so I came here thinking that I wanted to work on biological control, integrated pest management with entomopathogenic nematodes. But then I learned that that wasn't for me. It wasn't a love connection. I really wanted to do something that was related to evolution. And I switched and worked under Mike May, asking questions about dragonfly evolution. And since then, my research program has really evolved to focus on like who, what, where, when, why, like the basic questions of curiosity that I kind of started out with as a kid. How are insects related? When did insects diverge? And what drives some of the diversity that we see? So we've done a lot of work with international collaborators trying to get at that question of when overall insects first evolved. And I'm most interested in winged insects. And we know that winged insects from transcriptomic data, from molecular dating, are around 400 million years old. And this is really striking to me because, of course, we're just like a teeny tiny blip in the history of insects, in particular in the history of winged insects. But we have a really disproportionate impact. So another aspect of what I work on in my research program is really asking questions about insect decline and some of the drivers of insect decline. And if there's time at the end, I'll talk a little bit about that. So with colleagues, we formed a group called the 1000 Insect Transcriptome Group. And our goal was to use transcriptomes to try and resolve the phylogeny of the insects, a tree of life of insects. And so I thought I would use this just kind of as a map to kind of show the relative positions of what systematists think for insects, and then show you the coolest part, which is where the dragonflies are. So using transcriptomes, we find that basal hexapods, things like silverfish, firebrats, they're sister to the pterygoda or the winged insects. The non-holometabolous insects, which are the insects with incomplete metamorphosis, is paraphyletic. So it includes the group that I work on, the Paleoptera, the Polyneoptera, and the Paraneoptera. And of course, this is sister to the holometabola, the insects with complete metamorphosis. So since this early paper came out about 10 years ago, we've been working steadily, and we have a new manuscript that we're going to submit that's led by another former Rutgers alum, Paul Franson. And it has around 1,500 transcriptomes. It's a giant phylogeny, very computationally difficult to analyze. In general, the topology is about the same. But what's interesting is with this amount, a slightly higher taxon sampling, we can ask some more nitty-gritty questions about the timing of particular radiations for different orders. But I'll zoom in to the base of the tree, the earliest branching lineages, which are the Paleoptera. My postdoc advisor was Dave Grimaldi. He is like a very staunch mayflies were the first thing to fly guy. I think dragonflies were the first things to fly, and not just because dragonflies are cool, but because they have a bunch of adaptations that are unique, like indirect sperm transfer. So whether or not you think it's dragonflies first, mayflies first, or Paleoptera as sister to the rest of the winged insects, take-home message, dragonflies pretty early on, when there was nothing in the sky, was something that was probably dragonfly-like. So this is an interesting group to study because we're interested in flight, and they probably were among the first to fly. They're voracious predators in their juvenile stage, where they develop in fresh water, both in still water, lentic habitats, and flowing water, lotic habitats. And they can also be voracious predators in their adult stage, doing interception-style flight. They really vary in their size. I was talking with some students this morning about Nanothemis bella, the tiniest dragonfly. It's that you can get in New Jersey, to things that we call the giant helicopter damselflies, which are big wingspans. They feed on spiders, to really heavy dragonflies like the Pedalura in Australia that can actually take down birds. They have amazing vision, and part of their predation is so spectacular because of their vision, but they also use vision for color because they can recognize color in each other. And they have different styles of color. They can have bumps and rugosities on their skin, which we call structural pigmentation, but they can also have granules of pigment in their cells that can migrate up and down to give them a brighter color or a darker color. They use this for thermoregulation, but also for communication. They're global in their distribution. Some of them migrate really long distances across oceans, and some of them never leave the water from which they emerge, maybe traveling 11 meters in their entire life. And they have two penises. So they have a penis at the tip of their abdomen through which they make sperm. They take that sperm and they deposit it into a secondary genital apparatus at the base of their abdomen, near where the abdomen and the thorax connect. That's where they do sperm transfer, and it's indirect sperm transfer. And that's kind of unique. And we think it's perhaps related to an ancestral trait to have a spermatophore or a little sperm packet, which is what we see in the basal hexapods. So a lot of the innovations that we're interested in understanding for evolution of the insects started first in Odonata. So it's a great group to study. We often think of dragonflies and damselflies as being very ancient because there are these things that are called proto-odonates or pre-dragonflies and damselflies. And we know a lot about them from the fossil record. And the fossil record is complete enough for dragonflies and damselflies that we can kind of even infer some aspects of their behavior and their lifestyle. So these are what the proto-odonates look like, the Meganeura, Meganosoptera in the family Meganeuridae. These flew in the Carboniferous period around 350 million years ago. Each wing was about 37 centimeters. So that's a pretty big animal, and they had a beak. So they had a beak that kind of moved like this. They were predators too. The name we call these, though, are not dragonflies. So these are probably a stem lineage that's a close relative to a dragonfly, but they're actually their own group, which we call griffinflies. And there's a photo of my colleague from the Stuttgart Museum holding a life-size model of what one of these things may have looked like. What's neat is that we have fossils of what their juvenile stages look like, and their juvenile stages were aquatic. Dragonflies and damselflies have aquatic juvenile stages. They have a labial mass that shoots out. They grab their prey items. They have gills. We can see those features already in the Carboniferous in this Meganeura and Meganosoptera. So these are what the eggs of dragonflies look like. They either lay their eggs in plant material, which I'll talk about in just a moment, or they deposit their eggs on the surface of the water. Dragonflies and damselflies differ in what the nymphs look like. Dragonflies have kind of robust, stocky nymphs, and they breathe through gills that are in their bum. We call it the anal pyramid, these internal rectal gills. Damselflies tend to have these very thin filamentous or sometimes flap-like gills that are external that they use to breathe with. And they tend to be kind of delicate and not as stocky as a dragonfly. These are what the faces look like. So they have their mouth parts modified into a labial mask. In some cases, it's a flat mask. In some cases, it's like a scoop. And either way, they're mostly sit and wait predators. And they shoot their mouth parts out. They grab the prey items and eat it. And they eat each other a lot. They eat other insects, but they also eat vertebrates. So they're capable of taking fish and tadpoles when they're in their juvenile stage. So in general, we kind of, as systematists, like to just kind of sort things into piles. And that's a big part of what my job is sorting things into piles. And so using morphology and with molecular data, we've kind of sorted the order Odonata into 3 main piles, which we call suborders. There's around the same number of species of dragonflies and damselflies as there are all of mammals. And there are roughly 3,000 of them are damselflies, and roughly 3,000 of them are dragonflies. So the dragonflies are kind of stocky. They hold their bodies kind of low to the ground. They either perch on the ground or on veg, or sometimes they're flyers. They hold their wings out to the side when they're at rest. Damselflies have very slender abdomens, and they tend to hold their wings behind their back when they're at rest. Important to note, they cannot fold their wings the way that Neoptera can, because they don't have the basal sclerites to do that. So they don't fold their wings over their back. They just kind of have their wings positioned outward, which does make them vulnerable to predators. I'll just say that. And then there's this third suborder, which exists today as a single genus, Epiophlebia. It has 4 species. It's found only today in China, the Himalayas, and Japan. What's neat is genetically, we think it evolved from an ancient introgression, an ancient hybridization event between dragonflies and damselflies. It has morphological features of both. It has petiolate wings like a damselfly and stocky, thick bodies like a dragonfly. And then there's this extinct suborder called Cephalozygoptera that we've been describing a lot of fossils from recently. This is something that people for the longest time thought were just damselflies that had really squished heads. Their heads were always really squished. And like, oh, man, it's awful that every time a damselfly makes a fossil, their heads get squished. No, Bruce Archibald from Canada, he actually was the first person that described this suborder. And it turns out it's actually a unique synapomorphy, a unique shared derived characteristic, that they had really enlarged heads. And there have been some compression fossils that have been found where the heads are really intact. And you see that Cephalozygoptera, cephalo head, that it actually is unique to this suborder. It's one of their defining characteristics. So we've been working to try and, just as we tried to get a tree of life for insects, we've been trying to get a really comprehensive tree of life for dragonflies and damselflies. Around the same number of species as mammals. They did it for mammals, right? We have every single mammal sequenced, right? We can make a phylogeny with every single mammal on it. So we should be able to do it. Right? Well, that's true. Cryptozoa. Actually, I've got a Bigfoot in the slide. Just you wait. I've got Bigfoot coming up, but it's possible. This could be the order that we have everything for. So we have a large grant. We call it Genealogy and Odonatology. And our goal is to have species distribution modeling, ecological niche modeling for all the species, which we've already done, morphology for all the species, which we've already done, and then have sequencing data for all of the species. We think that once we have this phylogeny, we can test hypotheses people have been saying for around 100 years, that maybe color is what's driven diversity in this group, and maybe differences in dispersal ability and flight behavior has led to differences in this group. And just as a reminder, there's a huge amount of variation within just this order. Things that have structural color, things that have these migrating granules of pigment, things that are small, things that are large. Things that live in still water, things that live in flowing water, huge amount of diversity in this group. Now, the Zygoptera, I would say politely, are a mess, right? So we really have a lot of chaos. And I'm not going to talk a lot about Zygoptera in my talk today. I'm going to mostly focus on the Anisoptera or the dragonflies. But I'll just note that about 10 years ago or so, colleagues and I, we thought maybe there was around 13 or so families in Zygoptera. Now we think maybe there's upward of 50 of them. And people are kind of constantly redefining and describing. Most of the species discovery that's happening is in the Zygoptera. So if you're looking for a group that, you know, you could probably spend your whole life working on, Zygoptera is it, because there's not enough people studying it, and it's really chaotic. So there are several groups of dragonflies that we have had a hard time figuring out the relationships of them. And I thought I would just kind of walk through the ones that are the most problematic. And that way you can be keeping them in your mind when I show you what I think is the true tree of life for dragonflies. So the first is the Aeshnidae. These are the darners. I love this group. I actually named my daughter Aeshna after one of the genera that's in this family. They're fast flyers. Some of them are migratory. They tend to have these migrating granules of pigment. So a dragonfly like this could look bright blue in hot sun, and it could look purple in cold weather when it's just waking up. Whether or not this is sister to the rest of all the dragonflies has been hotly debated. Another group is the Petaliuridae or petal tails. Sometimes people group it with the Aeshnids, the darners. Sometimes people group it with another group called the clubtails or the gomphids. What's neat about the petaliurids is that people think that people don't think this, but there was one PhD student who got his PhD, by the way. This is what his PhD said. He got it at Berkeley in the 70s, so maybe that was why. But he did a lot of work in the Pacific Northwest. He was really interested in the genus Tenipteryx. Tenipteryx hagani is endemic to the western part of North America. And he spent a lot of time in the woods, and he thought that he had seen Sasquatch feeding on petaliurids. He argued Sasquatch was rare because their food was rare, and that if a human were to eat one of these Tenipteryx hagani, they would become a Sasquatch. And then the mystery, he defended his thesis, he got a prestigious postdoc at Oxford, but he disappeared. He went into the woods and then he was not seen from again. So there's like this lore in the odontology community about what happened to him. Yeah. So all of his writings, he kept journals. All of his writings are at the Smithsonian because the Smithsonian happened to buy a storage locker that had his writings in it. So you too can go and read his writings, but he drew lots of drawings that are quite beautiful biological illustration of this family and of the Sasquatch. But whether or not this is sister to the rest of the dragonflies, sister to the gomphids, or sister to the Aeshnids is a mystery. So the other problematic group are the gomphids or the clubtails. They're called this because the males in this group, the tip of their abdomen are expanded, almost club-like. They're not always club-like, but a lot of them are. If you are an angler or if you like to fish, these are your guys because a lot of trout, a lot of things that you are interested in catching like to eat gomphids. Gomphids are a really diverse group. There's over 1,000 species in this group. And uniquely, the females have lost their egg-laying apparatus. They've lost their ovipositor. So whether or not these are sister to the Sasquatch food or whether or not they're sister to the next group I'm about to talk about is debated. So the superfamily Cavalabiata, or Libelluloidea, as it's sometimes called, this is part of what I focused on when I was doing my PhD here. This is a really diverse group. If you've ever had to catch insects for taxonomy, chances are very good that the only dragonflies you caught were members of this group, because we almost always catch libellulids. They're very easy to catch, like catching fish in a barrel, as they say. So we don't know whether or not these are sister to the clubtails or whether or not they're on their own evolutionary trajectory. And so we wanted to ask this question, how are the odonates related? So as systematists do, we put some money, taxpayer dollars into sequencing a bunch of things. We looked at morphology, we looked at biogeography, we looked at behavior. One phylogeny that we made had around 500 or so genes. Another phylogeny that we made had around 2,000 to 3,000, 2,700 or so genes. And what we're finding is no matter the type of data that we use, we're kind of converging on what we think the relationship is among these three suborders, the damselflies, the dragonflies, and then third group that maybe was an ancient hybridization that only lives in China, the Himalayas, and Japan. We're actually really close to our dream, right? Our goal is to be like mammals, to have the whole entire order sequence. We have 3,066 of the 6,300 sequenced already. So like just to give props to our postdoc Lacey Newton, who really led this project. We're using a mixture of targeted enrichment sequencing. So we have between 100 to 1,000 genes, but our goal is to try and get 100%, to get every single species sequenced. And as I say, we already have behavioral ecological niche modeling morphological data for all of them. So just to kind of walk you through what we've done, we've used mayflies commonly as the outgroup. You want to choose an outgroup that's the closest member to your in-group that's not in your in-group. So as Paleoptera, those two are often recovered as Paleoptera, so they're our sister. Now, when I was in graduate school, there was a fellow named Andy Wren, and he had done a lot of work on this weird taxon. It's only found in southern Australia. It's called Hemiphlebia. It's metallic green. And the females do this weird thing. She holds her abdomen over her back when she goes to lay her eggs, and she just chucks them into the bog, right? And that's unusual because all dragonflies and damselflies either cut a hole in plant material and lay their eggs in plant material, or they tap their abdomen on the surface of the water rather than just chucking your eggs willy-nilly. And Andy said, I think this is sister to the rest of all of the odonates. It's neither Zygoptera nor dragonflies, nor Anisozygoptera. Well, no, we find Zygoptera being monophyletic, but we do find Hemiphlebia mirabilis being in the group that's sister to the rest of the Zygoptera. So it is a really early branching lineage. We don't quite understand this behavior, but we do know that this will probably go extinct in our lifetime because it exists today as just 6 small fragmented populations. There's a really large superfamily called Colopterozoidea. I have a tattoo of one of these. You guys have probably seen the ebony jewel wing. They're a member of this group. This family, surprisingly, we recover as being paraphyletic. It's not monophyletic. The superfamily. It has a bunch of different families in it. It's kind of what we call in systematics a junk drawer taxon. So if you don't know what it is, put it in Colopterozoidea. What we're finding, though, is that with this molecular data, we're able to use this plus morphology to do some revisions. I thought I would tell you about a couple of very cool members of this group. One is the family Polythoridae. This exists only in Central America and South America. They have metallic sheen on their wings. They actually mimic toxic butterflies. So they mimic Ithiomine glasswing butterflies as part of a mimicry ring to avoid predation. But look at their gills. So I mentioned damselflies have these little flimsy gills that stick out their bum that they use for gas exchange. And these look like maces, right? Like armory. If you look at them under a scanning electron microscope, they actually are like the medieval weapon, the mace, right? They look like that. And when you bisect it, it actually looks like it's filled with almost like upholstery foam. And you might ask, can they breathe with these? They cannot. When we look at their trachea do not penetrate their gills. And instead, they have gills on their abdomen. So no odonates, except for one other group I'm going to mention, have gills on their abdomen. They all breathe through either their bum in the Anisoptera or through these caudal lamellae or these abdominal, caudal gills. But for the Polythoridae and one other group I'll mention, they've evolved these innervated lateral gills, like what you might see in a mayfly. This is a cool group. I think they're very pretty. And this particular genus is one of my favorite for the damselflies, Heliocharis. But the reason why I mention these is if you look at these morphologically, they look like Cephalozygoptera. They look like that extinct suborder that has the weird smushed heads. These heads look more like traditional Zygoptera heads. But if you look at their wing venation would place them within that extinct suborder. So there's probably something interesting happening with this group. I wish we could get DNA from fossils, from compression fossils of that extinct suborder, but we cannot. But maybe the Cephalozygoptera is actually a close relative to the Dicteriatidae. So the euphaids, I got to see this for the first time last year when I went to Greece, to Cyprus. This is the other group that have lateral gills. If you look at their caudal lamellae, they look strange, right? They're not these little thin filamentous gills. They're actually gas-filled. And the caudal lamellae are filled with gas that kind of holds the abdomen so that it's floating on the surface of the water. So they kind of swim around with their bum touching the water. We're not quite sure why they do this because there are trachea that penetrate the caudal lamellae in euphaids, but they have these lateral gills. that are different and probably not entirely homologous in all of the features to the ones that you see in polythority, which is wild. And we don't know if you were to ask anyone about who's studying Euphaidae, the last family I'll mention in the superfamily is just one because we probably have all seen it. It's a classic New Jersey, right? You can go to the Pine Barrens, you can go to our site probably and see this summer, the Calopterigidae. And we do find this one family to be monophyletic, and it's very colorful and unique. Probably if you've had to take taxonomy and you caught a damselfly, you caught a little blue thing, right? Almost all the little blue things, the bluets, we call them, they're in the family Synagrionoidea. No big story here other than that the helicopter damselfly, which is a giant, it's a really, really big damselfly. They lay their eggs in tree holes or in bromeliads. They have a pretty big wingspan, long, thin abdomens. They're actually nested within these little blue guys, within the bluets, which is interesting. So on to the good stuff, which are the dragonflies. So that third suborder, the one that I said was maybe an ancient introgression, a hybridization event between damselflies and dragonflies, the Anisozygoptera, it's sister to all of the dragonflies. And this is what the nymph looks like. It's got internal gills. Its gills are in its bum. It has the same nymph shape as what a dragonfly would have. Interesting, right? So again, wings like a damselfly, body like a dragonfly, nymph like a dragonfly. So we're like, we got to look at some other characters. So we've been looking a lot at reproductive structures. Turns out their sperm is wacky. So their sperm is actually coiled around a little spindle. What has that? Dragonflies or damselflies? Neither. Neither. It has its own kind of sperm. So there's very few people that are working on this group. The material that comes out of China, there's one species from China, one species from Nepal. Those ones are virtually unstudied. We know a lot about the taxon that's in Japan. So if people are looking for a group that would be pretty fun to go to Nepal, this would be a group to study because we know almost nothing. So in the dragonflies, the Aeshnids, the darners, the one that we had a question mark about, we recover it as sister to all of the rest of the dragonflies with this other group that a former person who worked here, Frank Carl, described, the Ostropedaliidae. They're recovered as a monophyletic group. Now, that question about the Sasquatch food, the pedalurids, right? Where do they fall? They fall as sister to the clubtails. The reason why that's interesting is because people have spent whole careers trying to resolve that. And we have really strong support for the pedalurids and the gomphids being sister to each other. I had a graduate student who's in a job interview right now at UC Riverside, so let's give him good vibes for a tenure track position, Ethan Tolman, and he did a really deep dive into this family. What's neat is that this family exists only on the coast. So we have an East Coast one, Tacopteryx thori. We used to be able to get it in Stoke State Forest, but it hasn't been seen there in about 20 years. And every time people find new ones, we tend to build a condo on that site because that's what we do in New Jersey. So it might be locally extirpated. There's a kind of the West Coast one, which is the one that Perry Turner thought, or maybe he's turned into a Sasquatch when he was studying it. There's one on the coast of Chile, there's one on the coast of Australia, and there's one in Japan. So Ethan did a lot of work trying to look at the biogeography, and it turns out probably they were all together when the continents were more close together, and then there was a split into a Gondwanan, a southern hemisphere clade, and a northern hemisphere clade, Laurasian. I'm happy to talk more about their behavior because also those have nymphs that are terrestrial. So they don't actually live in water. They walk around on the ground and they're just in kind of damp areas. So if people are interested in behavior, we have a lot of data on that. So the gomphids, as I mentioned, they're one of the really species-rich group. There's around 1,000 species of those. And we think part of what drove the speciation in this group is that they've lost their ovipositor. So females don't have an egg-laying apparatus. They just kind of squirt their eggs out in a clump. The next group are the Cordula gasteroidea. These are really pretty. You can find these in Stoke State Forest still. If people go out to the Pine Barrens, you'll see these. And they have an elongated ovipositor. They have the opposite. And they're called darning needles or spike tails sometimes. The common name varies because they go like this in the water and the female kind of pokes her abdomen into the mud. And it kind of looks like a sewing machine needle going up and down. Now, the Libelluloidea is the super family that I worked on when I was in graduate school here. They're a really species-rich group, 1,500 species. And they also have lost their ovipositor. So we think the loss of the ovipositor maybe is driving species richness in this group. I'll mention the symptomicity because these are kind of a neat group. They're mostly found in Australia, but Mike May, a former professor here, he described this family and elevated it to family status in 2015, and we recover it as monophyletic. I have a couple of graduate students working on trying to revise some of the other close relatives. What used to be maybe 1 or 2 families is now 20. So they found a lot of morphological variation, especially in the genitals. So Rema Uchidike and Aaron Goodman have been working on this group. When I was in grad school, I had the opportunity to go and collect in Australia, and I found Pendithemis. But I also had a family member had gone to French Guiana, and he had collected Aschnosoma. And I looked at the nymphs, and I was like, Mike, these look exactly the same, but one's in Australia and one's in South America. How could that be? They all have these really long little projections that come off of segment 10. When we sequenced them, they recovered as each other's sister. And what's interesting is with this larger data set is we recover them as sister to the skimmers, which is going to cause a lot of people to be like this in dragonfly world, because people would have thought these were Horduliid-like or emerald-like. But no, they're probably sister to the Libellulids. Now, the Libellulidae was the thing that I was tasked with when I came here to Rutgers. I was so excited to figure out what was going on. When I started, maybe there was one family, the Libellulidae. There had been this guy in Germany who thought maybe there was 3 families. There was another person who thought maybe there was 5 families. And then there was someone else who thought there was like 13 families. And I, for my thesis, stood up in Blake Hall and I said, with my Sanger data set, I've actually solved this question. I think that it's probably just one family. And you could tell I'm probably foreshadowing here. So Harold Lech in 2007, myself, and Eric Pilgrim, we all kind of came to the same conclusion that probably it's just one family. All the subfamilies that have been described in the last 100 years were probably not valid, probably not monophyletic, except for one. There was one that was called the Libellulae that we all recovered. And if you were reviewer number 3, you might have noticed in our papers that along the backbone of the phylogeny of Libellulidae, there was no support. It was a polytomy, right? So while I stood in front of my committee and I was like, well, done. It actually was largely a polytomy for this family, right? And that's what Harold found in Germany. That's what Eric Pilgrim found out in Utah. So in the last, 15 to 20 years, we've really tried to get rid of that polytomy to resolve it. And the way that we do this as systematists is to sometimes throw money at the problem. So we threw more data at the problem. That was one of the questions, maybe having different types of data, genomic sequencing data, targeted enrichment or subgenomic sampling. Also maybe having more taxa, because I only was here for 5 years. I had 2 kids while I was here. I was a busy gal, so I didn't have time to sequence all 1,500, species. So maybe adding more taxa could have resolved this question. So one thing that we've done is we've really turned to collections, which are an invaluable resource. And we've used to think when I was in graduate school, especially if you were doing Sanger sequencing, that collections really weren't great, because if you had specimens that were older than 10 years, you couldn't get good DNA from them, so don't even try, right? But with some of the methods that we use now for targeted enrichment sampling, you can actually do better when you have older specimens, because it requires the DNA to be in small little chunks. Dragonflies that are collected nowadays, we put them in fingernail polish remover in acetone to preserve their color. It breaks the DNA into 650 base pair pieces. That's the perfect size for targeted enrichment. Actually, there's really a lot of great dragonflies that you can sample. So there's a lot of questions that go into using collections. I don't have time to talk about them all, but in the collections world, we have a lot of, history. The reason why we have a lot of collections at the American Museum, we have 23 million specimens, is in part because we could afford to, because we had robber barons and we had Teddy Roosevelt that was bankrolling this. We have specimens from parts of the world where researchers in those countries don't have access to those specimens, right? Only we have them. Who gets to use the specimens, right? These are heavy questions that I'd be happy to talk about afterwards. And I'll just answer this more humble question, which is there DNA in any of them? And the answer is yes. So we have a lot of collections that are from big famous expeditions that took place in the early 20th century. These are a pair that was collected in 1939. So we went through the collection and we tried to pick old specimens, and then we wanted to do a test. We did it at our museum, and we did it at a museum in the Netherlands called the RMNH. And we found that there was a lot of DNA in there. So the RMNH, their specimens were actually kept in temperature, climate-controlled rooms for a lot longer than ours. So there was less DNA yield in the American Museum specimens. But we got DNA from 1909. We got DNA from 1911. My granddad was born January 9th, in 1921. And so I picked a dragonfly from that day because I thought that would be cool. And sure enough, there was great DNA in it, and we're able to place it in the topology. Just getting DNA from a sample doesn't mean that you got the right DNA. You could just be sequencing bacteria. So if we actually look at it in our topology, the things that were very old, I've kind of colored with the red date of where they were collected, and they're grouping with their conspecifics and their congeners. So this really old DNA works in this phylogenetic context. Now, you all are too polite to notice something here. This is still a polytomy. So it was very polite of you to stare at this tree and say nothing. It's actually still not resolved, right? So if I'll zoom in a little bit so you can see these numbers, when you're doing bootstrapping, when you're looking for node support, if you have a lot of data, you almost always get 100%. So when we do subgenomic data or targeted enrichment, a number below 98 is like the pips, right? So this is a polytomy, right? So I just mentioned I spent about 20 years, trying to improve upon this polytomy. And what have we got? We've got a polytomy, same as what I found in 2007, Harold Lech, Pilgrim and Von Dullen. And just like what we found back then, we still get Libellulini, that one subfamily, as monophyletic. But this is exciting because what this is telling us is that whether we use transcriptomes, whether we use 500 genes, whether we use 1,000 genes, the fact that we're still getting a polytomy, that tells us maybe it's a hard polytomy. And that's a phenomenon in systematics where there's rapid radiation that takes place. And it's so fast that there's not enough mutations that accumulate on what we call the internodes, leading to the different clades. And that's very challenging for computer programs to resolve. But it doesn't mean that it's wrong. It just means that probably when there was this explosion of libelluloidea, of the skimmers, that just makes it mathematically very difficult to determine these relationships. So did more data help? No. Did more text help? We don't know. So we haven't sequenced every single libellulid. So I hold out hope that one, nanothemus extra one that we add might just be that combination of genetic information that kind of breaks, some of the branches and resolves it. So we're still sequencing some libellulidae, a little more to go. So we think we're coming close to having a true tree of life. For all of Odonata. We're going to be poised, we're on the precipice of being able to do what the mammal people can do, right? If we know everything about these taxa, we can really get into the nitty gritty. So when I was in graduate school, I went to a talk in Diener, and one of the Diener professors said, "Oh, you work on phylogenies. Why? What's the point? What are you going to do with your tree?" And I was flummoxed because I thought you just made a tree to make a tree, systematics for systematic's sake, right? And I didn't have an answer. And he said, you should figure out an answer before you graduate. And I learned, oh, wait a minute, you could use a phylogeny. We're using the phylogeny for something, right? That was the first time it had really occurred to me that this phylogeny could be used to test hypotheses, right? And that's basically what we do. Like, we're interested in systematics for systematic's sake, but then I want to use the phylogeny so I could evaluate questions and hypotheses people have had. So one of the questions we had was about this egg-laying strategy. I mentioned gomphids lost their ovipositor, libelluloids lost their ovipositor. So all damselflies have an ovipositor. It's like a little knife at the end of their abdomen. They cut a hole in plant material. They put their eggs in one at a time. It's very time-consuming, and their clutch size is restricted by the size of the plant in which they're laying their eggs in, right? Some dragonflies do that. They still have an ovipositor. But the two groups that lay their eggs just on the surface of the water, the gomphids and the libelluloids, those are the ones that are really species rich, right? There's 3,000 dragonflies, 1,000 are gomphids, and 1,500 are libelluloids, right? So almost all the diversity in Anisoptera are the ones that have lost this egg-laying apparatus. So you may have seen it. It kind of looks like this. They actually cannot distinguish between the surface of water and the surface of cars. So you might see them doing this in the surface of cars. If you go to Costco parking lot, if you're in downtown Newark in traffic jam, you'll see dragonflies flying over you. They're almost always members of this group that have lost their ovipositor. People have tested this to look at light refraction, and they actually really have an especially hard time with blue and black cars. But they're just wasting their reproductive output on your car. So we need a phylogeny to understand how many times this loss has occurred, right? If the gomphids and the libelluloids are sister to each other, that would mean it probably was lost once, right? The petalourids are the Sasquatch food, just as a reminder, and we recover the petalourids and the gomphids together. So the fact that these two groups that have lost their ovipositor are not each other's sister tells us that the reduction in the ovipositor occurred multiple times. And when you look at the morphology, you can actually see it because the ovipositor isn't just a single structure, it's 3 interlocking devices, gonapophyses, and the way that they're reduced is actually different in the two groups. So this exophytic oviposition question, we wouldn't have been able to answer it without a phylogeny. We use this phylogeny to ask this question. We know something about the timing of the divergence of these groups, and we think that probably what drove the loss of the ovipositor was the evolution of these amazing vertebrate predators, right? So once we see the rise, there was always fish that were eating them. There were always frogs, kind of from the early days, neobatrachid frogs at least. But then when we see the rise of pterosaurs, and then birds, and then later bats, this is when dragonflies got really crafty. They got much more maneuverable in their flight, and they lost their ovipositors. So we know, as I say, we know something about the way that dragonflies diverge because we have a phylogeny. We can use the phylogeny, we can calibrate it with fossils, and we can estimate the ages of these groups. So we think that for dragonflies, for damselflies, they're around 225 million years ago is when they diverged. So the Earth would have looked like this. And it would have been these like really basal synapsids, like really boring. This is not a T-Rex. This is not like the fanciest of the dinosaurs. They hadn't even evolved yet, right? And this is when we're seeing damselflies kind of diversifying and taking to the sky. Interestingly, we also find dragonflies to be around the same age. And that's when we think this introgression event happened in that third suborder, Anisozygoptera. So one of the other questions that we had about dragonflies and damselflies was about how their flight evolved. Eventually, we want to use this phylogeny to ask questions about the evolution of the type of dragonflies that are migratory, or the dragonflies that spend a majority of their time flying, we call them flyers, or the ones that spend a majority of their time perching, we call them perchers. We know that there's statistically significant differences in parts of their wing. In general, wing venation is a big part of the story of flight for dragonflies and damselflies, because if you have a lot of density of the wing veins, then the wing is very stiff. And if you have really sparse wing veins, like you see in some damselflies, the wing is very bendy and floppy, right? So there may have been selection for particular wing vein patterns. But it's actually more complicated than that because the ultrastructure of the wing, the actual physical characteristics of the wing vary with taxa. So if we zoom in with a scanning electron microscope, this is what the leading edge of the wing looks like. It actually is a bunch of spines. And those spines, people have shown, not in our lab, but in a colleague's lab, actually reduce sound when wind passes over the wing. So there's some strategy that has to do with their predation style flight when they're around their prey items. But these spines also kind of channel the air over the surface of the wing towards the hind wing, where eddies are formed. And so it has to do with the aerodynamics of flight. And there's also these sensory hairs, and these capilliforms and sensilla are sending signals to the brain, and that allows the dragonfly to control for pitch, for yaw, for the variation in movement that they need to do to be able to stay on course and in direction. And you probably noticed whenever you caught a dragonfly for taxonomy class, there's this little dot at the tip of the wing, the pterostigma. That pterostigma in the 70s, somebody thought maybe that acted as a counterbalance. So it was kind of like a weight at the edge of the wing, and that would prevent the cord, the wing, from actually vibrating too much when there was fast style flight. If you look at the close-up of it, some of them have these kind of bumps and ridges and rugosities, and there's a lot of variation in what it actually looks like. It turns out there is a statistical significant difference in the weight and in the overall area of the pterostigma between perchers and flyers. And if you were to kind of cut, like with a scissor, kind of bisect it, and look at what it looks like. In perchers, it looks like very spongy, almost again like upholstery foam. And in flyers, then it's very, very, very thin and compressed. And we think this has something to do with hemolymph flow through this part of the wing and also with the total mass that's at the tip of the wing. So I'll just end by saying, doing this work, whether we're looking at flight, whether we're looking at reproduction, whether we're looking at the timing of dragonflies, only just has made me love dragonflies even more than when I started here at Rutgers. And so I'm particularly concerned about whether or not they're going to make it, right? We know that a lot of insects are declining. We know from the aquatic insect working group that I lead as part of this insect research coordination network grant that I have, that all aquatic insects are doing quite poorly. Some studies have looked at dragonflies, but most haven't, because people have often looked at EPT, Ephemeroptera, Plecoptera, and Trichoptera, for which we have a lot more data. But in general, the trends are steep decline. Last Thursday, Eliza Grames and colleagues, they looked at 76,000 records of butterflies just in North America in my child's lifetime. So from 2000 to 2020. And the average was a 22% loss. Things like the cabbage white butterfly, 54% loss. And some taxa were 98, 99% loss. We see this already for dragonflies in Europe. They've done a lot more work assessing dragonflies using the IUCN Red List. And it's a similar trend. The average is around 25 to 30% loss in Europe for odonates, around 50% for many of the specialist taxa and things that are extirpated. So are they going to make it? I don't know. They've been around a lot longer than most of the other insects. But freshwater is a limiting resource. All life on the planet relies on freshwater. And what do? We throw a bunch of junk in it. Plastic, garbage, pollutions. We roll back EPA protections. If we want dragonflies to make it, and we should, they're important food sources for a lot of animals, and they're top predators. They're people who think I'm at Rutgers still call me all the time every summer asking, when's Rutgers going to release those dragonflies to get rid of the green head flies in my yard? And I'm like, I don't work there. But like they do important service. They do important services for us, right? So it should behoove us to want to be advocates for the dragonflies. So if you're interested in finding out more about what we're doing, I'll mention a couple of the dragonfly teams that I'm part of. One is the Geo group that's looking at the evolution and ecology of dragonflies, and one is the Dragonfly Morphology group. I'd like to thank my graduate students and postdocs with whom I work. But then I'd also like to thank Mike May, who was a professor here for many years. He started at Rutgers entomology in the 1970s. And I was fortunate enough that he was my major professor. He died in 2023. But his love for dragonflies really is what got me going and what has kind of sustained our odonatology community. It's a fun group, and it's a fun group because Mike made it a fun group. So if there's time, I'll take any questions.
So maybe it is